Okay, so during the break, I went and pulled out my textbook and turned to page 341, which has the genetic code table in there so that I can answer the second part of the question. But let's talk about the first part of the question first, or the first question first. Describe how a polypeptide can be secreted from a cell reaches the endomembrane system. So remember, that's the signal particle and the signal peptide at the beginning of the polypeptide sequence. So any polypeptide that is destined for the endomembrane system has a signal peptide at the beginning of the sequence. And so that is recognized by the SRP, and the ribosome complex, including the messenger RNA and the polypeptide, are escorted down to the surface of the endoplasmic reticulum. They anchor to the translocation complex, which opens up and allows the polypeptide to enter into the lumen of the rough endoplasmic reticulum, from which it will be packaged into a transport vesicle and moved on through the endomembrane system. So that's how it takes place. So draw a simple diagram of a tRNA with an anticodon sequence of CGU. So if we use the textbook's example, let me bring up the pen. We could do it this way. And it would be 3' C, G, U. Now, if you look at the code table, I know the question says, what are the two codons it can bind to? In fact, it should be able to bind to 4, because there are 4 codons that begin GC, because C is going to be complementary to G, G is going to be complementary to C, and U is going to be complementary to A. But if we take a look at the genetic code, we find that there are 4 codons that begin GC. So the other one would be GCG. That would be 2. Then it could be GC. Well, it can't go to GCU because U is not compatible to U. And it's not going to be compatible to GCC. So yeah, there's the reason why it's only going to be compatible to 2 of them, because the U is only compatible to A or G, not going to be compatible to U or C. So that's the reason why it's going to bind. This will be the 5, oops, I don't want the eraser. I do want the pen. This will be the 5' end, and this will be the 3' end. This will be the 5' end. This will be the 3' end. And the amino acid, alanine. But there are 4 codons for alanine. Okay, so the final section, and we are at the final section, talks about the effect of changes in nucleotide sequence, otherwise known as mutations. Now, the most common mutations affect single nucleotides, and those are referred to as point mutations. But because if you change the nucleotide, a single nucleotide in one of the 2 strands in the DNA, you are automatically going to change the nucleotide that is complementary to it in the other strand when the DNA is copied. So while we talk about base pairs being single nucleotide changes, we actually, or point mutations as being single nucleotide changes, we actually refer to them as base pair changes, because as soon as you change one nucleotide, you are going to change its partner, its complementary partner on the other strand. So you're going to change both nucleotides in that pair. So a point mutation is sometimes referred to as a base pair change. These are the most common types of mutations. But in terms of the effect on the polypeptide, it's going to come down to which of the nucleotides in the codon is going to be affected. So here is the effect of the most frequent mutation associated with sickle cell, the sickle cell condition. So the normal sequence for the beta globin polypeptide, which is the one that gets affected, and this is residue number 6 or amino acid number 6 in the beta polypeptide. The coding sequence, and remember the coding strand or the template strand is the one that reads 3' to 5', is CTC. That gives a codon in the messenger RNA of GAG, which in turn codes for the amino acid glutamic acid, one of the acidic amino acids. The mutation involves the change of the T to an A, what is referred to as a substitution mutation. But notice that it also affects the nucleotide in the non-coding strand. So the A that is in the non-coding strand is now a T. So the new sequence is CAC. That gives us a codon in the messenger RNA of GUG, and that is a codon for valine, which is a non-polar amino acid. So we've gone from an acidic polar amino acid to a non-polar amino acid. And that's the reason why this has such a significant effect on the final folding of the beta globin, because you've changed the chemical nature of this area of the polypeptide. You've gone from a polar acidic region to a non-polar region, and that changes the folding of the polypeptide and leads to all of the phenotypes that are associated with sickle cell. Now, the reason why this is so significant is because it's changing one of the non-wobble nucleotides. It's changing the middle nucleotide of the codon. If it had been changing the third nucleotide, the G, it may not have had any effect at all. But because it's changing the middle nucleotide of the codon, it is changing the amino acid. Now, there's one other thing that I want to show you here. A trick that you can use if you're asked to do, say, a transcription thing, or if you're of a DNA, or if you're asked to figure out anticodons on a tRNA, there are a couple of tricks that you can use, and this diagram is really good to show you that. So, what if you were asked, what is the codon in the messenger RNA that this sequence would give you? So you know that the gene sequence is CTC, but the messenger RNA sequence is going to be GAG. But notice that that's exactly the same as the complementary DNA. So even if you did not remember your base pairing rules, you could simply look at the non-template strand, and that would give you the messenger RNA sequence. Of course, you would have to remember, however, that in the messenger RNA, T is replaced by U. So we see that here. GTG becomes GUG, using the same idea. Okay. Now, if you were asked for the anticodon, remember the anticodon is going to be the complement of GAG, which is CTC, or in the case of the RNA, we wouldn't put a T in there, we would say CUC, that would be the anticodon. So you can use your knowledge of the base pairing rules, you can use your knowledge of the complementary nature of the nucleotides to figure things out without necessarily having to spend a whole lot of time writing things out, because if you remember that the complement of the template strand is the messenger RNA sequence, but the complement of the messenger strand, the template strand, is also the non-coding strand, you can figure out what the messenger RNA is going to look like, so long as you remember to replace the T's with U's. If you're asked about anticodons to a codon and all you have is the DNA sequence, well, if you look at the nucleotide sequence in the template strand, that is going to be the same as the anticodon sequence, so long as you remember to replace the T's with U's. Just some simple tricks to answer some questions that you may find in the exam. Just putting it out there. So when we start thinking in terms of point mutations, the two most common types of point mutations are substitution mutations, which you've already seen. That's the sickle cell mutation. The other one is referred to as an insertion or deletion mutation. Of the two types of mutations, these are the most significant. And the reason why is because these always change the reading frame. Always. And here's the reason why. Using a sequence of letters. And I told you I'd show you an example of this using a sequence of letters. So there's a sequence of letters. There is a message in there, and it's in 3-letter words, not an accidental, deliberate choice. And all you need to do is figure out what the 3-letter words are, and I'll simplify that by highlighting them for you. There it is. The cat ate the rat. There's the sentence. There's the message. Now, watch what happens when I take a letter out. So we'll do the same thing again. We'll assume that it's the same message, and we'll start making 3-letter words. So, uh-oh. We've changed the reading frame. We've changed the grouping simply by taking a letter out. And so the message has become nonsense. It has no meaning as far as the original message is concerned. If I was to add a letter in, you can hopefully realize that you would get the same thing. If I replaced a letter, so if say I took this T out and replaced it with an E, which would be the equivalent of a substitution mutation, yes, this particular word no longer makes sense, but the remainder of the message, the cat, the rat, is still there. And you could probably figure out what this was. The vast majority of the message is still there, and you can make sense of that. And so substitution mutations typically don't have a huge effect, particularly if they affect the wobble base. But insertion mutations and deletion mutations, they always have an effect because they always change the reading frame. And by changing the reading frame, what's referred to as a frame shift mutation, you create a message that no longer makes sense. So in the textbook, you have this diagram, and I would strongly suggest that you take a look at this diagram because it shows you everything that you need to know about the nature of mutations. So here is our DNA, and I can't see the template strand under this, so I'm just going to slide this down for a minute. So the template strand, you can see here, remember it's read 3' to 5', so it's this strand reading across, that's our template strand. Here's our messenger RNA, and here is the protein sequence that translates from that. So down this side, we have substitution mutations. Down this side, we have insertion, deletion, or deletion mutations. So here is the first substitution. We change the G to an A, which changes the C that was here to a U. Well, GCU and GGC happen to both be codons for glycine. So there's no change in the amino acid sequence in the polypeptide. And as a consequence, this is known as a silent mutation. Why silent? Because while there is a mutation here, there is a change in nucleotide sequence. It has no effect on the amino acid sequence in the polypeptide. So it has no effect on the polypeptide. It's silent. It has no effect. This time, in this one, we're doing the substitution in the first nucleotide of the same codon. So the C now becomes a T, which means the G now becomes an A. AGC is a codon for serine. And this now becomes a missense mutation because you've changed a single amino acid, but the remainder is intact. So technically speaking, the sickle cell mutation is a missense mutation because it's changing a single amino acid in the sequence of amino acids in the polypeptide. It just unfortunately happens to change a key amino acid in a very important location in terms of the final three-dimensional shape of the polypeptide, and therefore creates a huge issue. But the vast majority of missense mutations are not that significant because you're only changing a single amino acid. The vast majority of the polypeptide remains intact, and therefore it is likely to be more or less functional. The sickle cell hemoglobin happens to be an extreme example of where that is not the case. If the change results in the loss of the polypeptide, you create a stop codon, for instance, so that there's no polypeptide. This is referred to as a nonsense mutation. Why nonsense? Because you're not getting a polypeptide. It makes no sense, nonsense, no polypeptide. Now let's look at these insertion deletion mutations. And you can imagine, based on what we saw with the example that I gave you, things are going to be pretty interesting here. So here is a insertion mutation where a single nucleotide has been slotted in between the first and the second codons in the sequence. That's changed the second codon to a UAA instead of an AAG, and UAA happens to be a stop codon. And so this frame shift mutation has led to the loss of the polypeptide. This is the equivalent of a nonsense mutation, but it is referred to as a frame shift mutation because you've changed the reading frame, right? You've changed the way we frame out the nucleotides in groups of 3 as codons. Here we have taken out a nucleotide. So what should have been an AAA has now become an AAC because of the loss of that A. That means instead of a UUU, it now becomes a UUG. So we changed this amino acid. But what's happened now is we've lost the stop codon. We don't know where the next stop codon might turn up, if one actually exists in this sequence now. So again, it's a frame shift mutation. It has changed the whole outcome of the polypeptide, particularly because it's close to the beginning of the polypeptide. We're now going to get a totally new polypeptide that may or may not be functional. Again, this is why these insertion and deletion mutations are so significant, because they really do change the outcome. because they create that frame shift by altering the reading frame. Here's the one situation that may not be quite so critical. If you either delete a whole codon or insert a whole codon. Very rare, but has been known to happen. Again, this would be kind of equivalent to a missense mutation because the vast majority of the polypeptide is intact. You've just lost a single amino acid. So this may actually still be a functional polypeptide. What you need to remember, however, is that in each of these cases where we've had these changes in nucleotide sequence, we've created a new allele. So even this one, which has created no change in the amino acid sequence, is still considered to have created a new allele because it's changed the nucleotide sequence. And that's the importance of mutations. Mutations are the only source of new alleles. And remember, it's alleles and the genetic variation that they represent that are the secret to the success of organisms because of natural selection and evolution. Natural selection and evolution only work with genetic variation, which means they require the presence of alleles. And the only way to create new alleles is through mutation. So even though some mutations, and maybe we could say most mutations, are of zero value because they're either negative or neutral, periodically we may get a mutation, a new allele, that is significant and has benefit. And that opens the door for natural selection to favor the transmission of that allele and for the increase in the frequency of that allele. In other words, the organisms, the population where this allele exists will start to evolve in the direction of that allele and whatever phenotype it happens to be coding for, because it's a favorable phenotype. So we know that mutations can occur spontaneously and do occur spontaneously as a result of the errors during replication. Remember, a replication is very, very accurate, but not perfectly accurate. One error in every 1 billion nucleotides, approximately. Recombination can create mutations because you can lose sequences. Remember the idea of deletions and inversions and duplications as a result of non-reciprocal or misdirected exchanges during crossing over, and also by errors during repair of DNA. If there's no template for the DNA polymerases to use to add in the correct nucleotides during DNA repair, they're just going to add in random nucleotides. Or if the repair mechanisms fail to repair damage to the DNA, then again, the DNA polymerases are just going to add in random nucleotides as they try and read through the damage, because they're not going to be able to recognize the nucleotides and add in the correct nucleotides. All of those are going to result in mutations. And we also know that some physical agents and chemical agents can cause mutations. And one of the things that we've discovered about these is there are only certain types of mutations that can occur. So there are only certain types of substitutions that can occur. There are only certain types of deletions and insertions that can occur. And so our ability to be able to recognize the changes that have occurred in DNA over time as a consequence of different types of mutagenic agents, and to be able to recognize where those changes have occurred, has allowed us to extend our ability to use DNA information to look at relatedness, particularly when we start trying to look at the evolutionary history of organisms and which organisms are related to other organisms as a consequence of descent from a common ancestor. We can look at the changes that have occurred in the DNA in each of those two species and look at what changes have occurred and what the common ancestor sequence might have looked like as a consequence of that. And we can also use that as a way of determining when the two new species diverged from their common ancestor. It starts to let us build a better story of the evolution of life on the planet. Lots of interesting things that we can do with this information. So this brings us to the last slide for this presentation, and therefore brings us to the end of the semester. And it brings us back to the original question that we could have asked at the beginning of this chapter. What on earth do we mean by a gene? Remember, we had the gene hypothesis that was proposed by Beadle and Tatum, and then was edited to take into account what was learnt about proteins and polypeptides. But now we have to really go back and start the whole process again and ask ourselves, what really is a gene? Because what we've discovered is that it's not necessarily what we thought it was. A gene does not direct the assembly of a single polypeptide. We can't even make that statement anymore. We have to acknowledge that it codes for polypeptides, but it may not necessarily code for one polypeptide because of the exon story. It may not necessarily lead to the generation of a polypeptide because we've got all of those RNA molecules that we've now got to account for, like the tRNAs and the little RNAs like the what are known as iRNAs, interfering RNAs, and snRNAs, and other ones are known as SNPs and SNRPs. SNPs are found in the spliceosome, SNRPs are found elsewhere. These are all tiny little RNA molecules. They are the product of transcription, so they are the product of a gene. So in the end, probably the best that we can say about a gene is that, and this is the statement at the bottom, a gene is a region of DNA whose final product is either a polypeptide, not a specific polypeptide, but just a polypeptide, or an RNA molecule. That's probably the best that we can say if someone asks, what is a gene? More to come, I guess, as people think this question through a little bit more. Before we go, 2 more concept check questions. So let's take a look at these. What happens when a single nucleotide is lost from the middle of the coding sequence of a gene? Well, that results in a frame shift, right? That's a frame shift mutation. That's a deletion mutation. So that means we've got a frame shift mutation that changes the reading frame, the way the nucleotides are grouped into triplets as codons, which is going to change the amino acid sequence in the protein downstream from that deletion. And we can actually use the term downstream here, because remember, downstream means the direction in which we're reading the DNA and the direction in which we read the messenger RNA. It's downstream. So downstream from the mutation, the sequence of amino acids is going to be altered as a consequence of that deletion. So here is an opportunity to do some transcription and translation type work. The normal sequence of a template strand is 3' TAC TTG TCC GATA TC to the 5' end. It gets mutated, and it's a substitution mutation. So TAC TTG TCC, here's the substitution. The G has now been changed to an A, so it becomes AAT ATC 5' end. write out the double-stranded DNA for each, the resulting messenger RNAs, and the polypeptide sequence. So let's do the double-stranded DNA. So we've got, let me change it to red first of all. So we've got T, A, C, T, T, G, T, C, C, G, A, T, A, A, T, A, T, C. Now I'll put the other strand in here. The mutation T, A, C, T, T, G, T, C, C, A, T, A, T, C. So let me change the pen now. So T is A is T, G, A, C, A, G, G, C, T, A, T, A, G. And this would be 3 prime, this would be 5 prime, and this would be 5 prime, and this would be 3 prime. So now let's take a look at the mutation. Again, this is the 3 prime, this is the 5 prime, so this is going to be A, T, G, A, C, A, G, G, T, T, A, T, A, G. This is the 3 prime, and this is the 5 prime. Now, the messenger RNA sequences, what are they going to be? Well, remember, the messenger RNA is simply going to look like the non-coding strand where the T's have been replaced by U's. So let's change the color again. And the mRNA. And so it would be A, U, G, A, C, A, G, C, C, U, A, U, A, G. So that's messenger RNA there. Messenger RNA here is going to be A, U, G, A, C, A, G, G, U, U, A, U, A, G. So now we need to break those up into the codons. So it's going to be an AUG, an AAC, an AGC, CUA, and a UAG. Here it's going to be AAG, AAC, AGG, UUA, UAG. Now, all of them are going to be the same. Yeah, this should be an AGG. I'm not sure why I put a C there, but that should be an AGG, as is this one. So all of them are going to be the same. So this is going to be methionine, I know that. That's the AUG. Then the one codon that's going to be different is the CUA versus the UUA. The UAG, that's a stop codon, if I remember rightly. Let me get out the code. So it's page 341. So UAG is a stop. So that I was correct. That's a stop. And that's a stop. CUA. CUA is a leucine. Whereas UUA, well, UUA is also a leucine. Leucine happens to be one of those amino acids that has 6 codons, and both of those codons happen to be for leucine. So AAC is the only one that we've got left. AAC is aspartame. Well, I was wrong. I've left out one, the AGG. So AGG is arginine. So there we go. Now, would I ask you a question like that? Perhaps not as complicated as this, but I might ask you to do a translation or a transcription or something like that. And no, you don't have to memorize all the genetic code. I will provide you with all the information that is required for you to answer the questions. It will be included in the question in the exam. So don't panic. Don't feel like you have to memorize the genetic code and codons and things like that. You don't have to, not required. Only researchers go to the trouble of memorizing all of those because they use them all the time. And that's why they tend to memorize them, because they're using them all the time. You will get the information that you need to be able to answer the questions if I ask you to do something like this in the exam. Anyway, that brings us to the end of the semester. Thank you very much for participating. I hope you have found this interesting, and once we got organized with the narrations, breaking it up into chunks, doable, and that you were able to find your way through all of the narrated presentations. Yes, I realize that even some of the sections got a little bit long, but I tried to make sure that none of them were more than an hour long, and certainly substantially less than that. We have two more exams. We've got an exam this weekend, and then we've got the final exam next week. In the meantime, if there are any questions, concern, or comments that you might have about anything that I've covered today or anything throughout the semester, please feel free to come and see me during office hours. I will be having regular office hours during this week. Next week, because it's finals week, I won't be having regular office hours, but I will be accessible through email. And if you send me an email, I'll either reply to you if I can answer the question via email, or if I think I need to talk with you and perhaps draw diagrams and things like that with my tablet, then I'll try and set up a Zoom meeting with you to get that taken care of. So, unless I see you again, good luck on the remaining exams, and I'll see you around perhaps. Bye.